I just wanted to, well, first, welcome. So great to have you all here. I just wanted to remind everybody that the World One Health Congress is coming up. This year it's going to be in Lisbon, and abstracts are due on March 13th, I think. I'm on the scientific committee, so my job is to try to plug it. But I just wanted to let you guys know that we're also going to push that out. But if anybody's interested in learning more, there's a QR code to get some information on that. And then I'm really happy, thanks, Kelly, to be able to introduce Mark Zabel. Dr. Mark Zabel, I learned last night, came here 22 years ago. And he's a professor and associate director of the Prion Research Center in the Department of Microbiology, Immunology, and Pathology. And he was the previous research associate dean also for the College of Veterinary Medicine and Biomedical Sciences. He earned his PhD in experimental pathology from the University of Utah School of Medicine and completed a postdoctoral fellowship with Dr. Adriana Guzzi at the Institute for Neuropathology at the University Hospital of Zurich, Switzerland. I know you do a lot of stuff, but the work he's going to talk about today focuses on prion immunology, pathology, diagnostics, and therapeutics. Take it away, Mark.
Thank you. Thanks for coming, everybody. It's really nice to be here. Thanks for the invitation, and thanks for the serendipity too, because I was actually scheduled, as you probably know, for next month, and I had inadvertently double booked myself. Jen, see you. And my former student, who's now a professor at the University of Wyoming, invited me to give a talk, and I didn't check my schedule, and it happened to be on the very same day. So I am so glad that I could present today as an alternate. So thanks for coming. And I am going to talk today about a project that is 10 years in the making, but only funded the last year and a half. Right. So this has been projects that we did opportunistically, and I'll go into a little bit more about that. But one of the things that we're interested in about chronic waste, well, let me back up and just say, I always want to start with a thank slide in case I run out of time. Right. So these are the people that actually did all of the work. I'm going to talk about 3 different projects today, all about environmental prion contamination. One is going to be about our surveillance efforts in the Navajo Nation on the reservation in the Four Corners area, specifically the northern Arizona part of the reservation. And that was in collaboration with this gentleman here, Dr. Gilbert John, who I worked closely with when I was Associate Dean for Research. He is the Assistant Dean for Research. And so we started a project that became USDA funded to survey the Navajo Reservation for several different infectious diseases. My part was looking for chronic waste and disease on the reservation. And then I'm going to talk also about our efforts in the Rocky Mountain National Park. And that was, there's lots of people that have worked on this project over the years, mostly graduate students. And this really started as just a team building exercise for me and my lab to go backpacking in the national park. But we had done some work with the National Park Service on the eastern slope, tagging and sampling elk in a partnership with a GONACOM study for fertility control. But we partnered with them to take samples and look for CWD prevalence. So we had already established a relationship looking at CWD in the park on the eastern slope, but nobody had looked at the western slope where many of the elk go in the summertime because it's cooler, it's higher, there's less people. And so we started, opportunistically, just as we were going through the park, picking up samples, right? Environmental samples, typically feces, plant samples, soil, and some water if it was nearby. So easily, 8 to 10 graduate students have worked on this over the last 8 to 10 years. And then we also had several summer students working in the Navajo Nation to help collect these samples. This is extremely hard work. The reservation is huge, and it's a completely different environment to sample compared to the Rocky Mountain National Park, where we can backpack, we can go into the park, and it's hard to take a step without stepping on some elk feces, right, or moose droppings. The Navajo Nation is completely different. It is huge, millions of square acres, and so that required renting all-terrain vehicles. In that top right corner over there, you can see a cool shadow of us in the ATV coming back from sampling. And so we've had veterinary summer students work on these. We've had international students like Takashi from Hokkaido University. Josie Beauchamp was an undergrad from Fairbanks, Alaska, that worked on this project too, and my lab manager, Lisa Cofield, who did most of the laboratory work that I'll show you today too. And then the third vignette I'm going to tell you about is a new collaboration we have with the Arkansas Game and Fish Commission to look at CWD contamination in Arkansas, and I'll show you where that is in just a second. But just to orient yourself, about prion diseases. I feel like when I talk about this at CSU, it's not as foreign as when I have to talk about it elsewhere. Anybody have never heard of prion diseases before? Okay, good. I've got somebody that I can try to educate. Okay, so prion diseases are protein misfolding diseases, which means that unlike most infectious disease agents that replicate using a genome, either bacteria, viruses, mold, any other living organism. Prion diseases, according to the prion hypothesis, asserts that the infectious agent is just a misfolded form of a normal protein that we all express. And if I kind of go, this is sort of my working hypothesis for what happens. So you start with the normal prion protein. All mammals express this and a lot of different cell types. And then, this is theoretical. It's been a lot of talk about some sort of unfolding intermediate. And so I just have that labeled here. I have another really cool project where I think this is a real thing, and I've got data to show that, but it's beyond the scope of today's talk. But a lot of people have proposed an intermediate unfolding event, which then leads to this aberrant misfolding to an infectious prion form that we call PRP-SC. The SC is for scrapie. Scrapie is the first prion disease that was identified over 2 centuries ago in merino sheep in Spain. But once it forms that prion form, it becomes metastable. So it converts from a mainly globular, beautiful kind of alpha helical shape, a typical protein that you think, performs a function, to this mess, which is basically a beta sheet. I kind of describe it as turning it from like a balloon, the beautiful 3-dimensional shape, to like a flat 2-dimensional, like a bathroom tile, right? So this would be a monomer of those bathroom tiles, and then those monomers start stacking up onto each other to form a fibril, and then those fibrils kind of twist together and then form those amyloids. And those are the things that invade our brain and cause prion diseases, okay? And you can see this biochemically on a Western blot. If you try to use proteinase K to digest away the protein, normally it goes away. But if it's become a prion, it becomes PK resistant. It cleave off just a little bit of protein, so that makes the bands shift down because it's a lighter form of the protein. But this is typically what you would see, right? Normally, you digest away the normal form, the prion form sticks around, okay? And then chronic wasting disease is a specific prion disease that affects wild free-ranging cervids. So those include deer, elk, moose here in Colorado, but also, caribou, reindeer, any cervid species in North America. What I'm showing you here is a time-lapse photo from starting at 2004 to 2024, showing the spread of CWD in North America. It's also been identified in South Korea, thought to be started by transporting infected elk from Canada to South Korea. And it's most recently been discovered in Nordic countries. And there is no known link between CWD in North America to CWD in the Nordic countries. And that has really kind of changed how we think about the etiology of CWD. And it does allow me and other people at CSU to exhale a little bit because, as you might know, the first cases of CWD were found here or just on the Foothills campus in some sheep pens. It was thought to be maybe caused by deer and elk jumping into those sheep pens, and some of those sheep may or may not have had scrapie, and then the cervids jumped back out of the pen. And now we've had a trans-species event, and that's historically what people had assumed was causing CWD to spread, a jump from scrapie to CWD. Now that we have this new set of cases in Nordic countries that have no known connection to North America, and additionally, a lot of experimental evidence from many people in the field, including my own lab, that shows that there does seem to be a spontaneous component to CWD. And that's not surprising. We've seen that in human prion diseases too. The most common prion diseases are, Creutzfeldt-Jakob disease in humans, not very common, a little less than 1 in a million. But most of those cases are sporadic, right? So they're spontaneous. So we do feel a little bit better about not contaminating the entire planet with CWD, starting in Fort Collins. But how does it spread, right? So it is a wildlife disease, and in terms of prion ecology, prions, as I had shown in that previous slide, when you digest it with protease K or any other proteases, it's extremely resistant. It's also extremely resistant in the environment. So these prions can stick around for a very long time. And we think, and there's a lot of experimental and epidemiological modeling data to suggest that this indirect component where you have prions shedding in the environment from excreta, including feces, urine, saliva, blood, decaying carcasses, either from gut piles from hunters or just naturally dying cervids afflicted with CWD, can deposit prions in the environment that can then be transmitted indirectly to naive cervids as they rut or they forage plants that might have been contaminated through excreta exposure and the like, right? So we know that we can't explain these high prevalence rates for CWD in mathematical models without invoking an indirect component as well, right? So there's very strong evidence that CWD in the environment is a critical linchpin in the life cycle and spread of CWD. Okay, so we're going to start talking about our efforts at the Navajo Nation. This shows you our main sampling area here as an inset from this larger sampling area. So this is the Four Corners area. This is where we spent most of our time looking for CWD. Our last trip, though, we went into southern Utah and the northern part of the reservation and did some sampling there as well. Yeah, okay, and I'm just showing you. And then in the Romo locations, these are the areas that we've sampled over, I think we're on our 10th season now of collecting here. But what's really interesting, and I'll come back to this, I just wanted to introduce where we've sampled. This pink area is the East Troublesome Fire burn scar that happened in 2020. So we've been collecting samples in the Rocky Mountain National Park since 2017, right? Including areas where the scar occurred, right? So we've got samples there before and after the burn. And this is a central question that I'm going to address today. And that is, what is the effect of wildfires and maybe more importantly, controlled burns or prescribed burns on the CWD load that we find in the environment, right? So we've got this incredibly unique sample set that I think really pushed us over the top to get USDA funding, right? Because now we can look not just in the burn scar area before and after, but we also have areas that have never been burned. And so we can compare all of these different samples to samples that we collect after the burn and see if there's a difference in, say, the rate of positivity from those samples that we're collecting. Okay, so we'll come back to that. And then the Arkansas field collections, we don't do those field collections ourselves. Our partners at the Arkansas Game and Fish Department, Corey Gray and Ellery Lassiter, have been sampling in this area called the Gene Rush Wildlife Management Area. And it's just south of the Buffalo River National Preserve, where they have some of the highest CWD prevalence rates in the country, in the world, really, right? And so they are sampling this area because this is an area that they're considering to do controlled burns after we sample before those controlled burns. So a kind of a real life experiment in situ. We're also, and I will show you data of this, modeling prescribed burns in our laboratory using a small furnace and experimentally contaminating plant and fecal samples and then burning them at specific temperatures for specific durations, and then using a really sensitive assay that I'll talk about next to see if we've been able to eliminate it or at least reduce the prion titers after burning. Okay. And so the technique that we're using for all three of these projects is called real-time quaking-induced conversion, or RT-QuIC. And it's basically an in vitro system to amplify a prion seed if it's present by combining it with a substrate, which is normal PRPC, the normally folded form. If there's a prion seed present, it can seed the templating and misfolding of that PRPC to more of the prion seed, right? And then we can either PK digest and do a Western blot like I showed you before. That's low throughput. We've since changed this to a 96 volt plate format and a thioflavin T. So this is a planar molecule that when it intercalates into amyloids will fluoresce in the green wavelengths. And we can read that on a plate reader, okay? So that really increased our throughput. It increased our throughput further because obviously, you know, when we're looking in the environment for positive CWD samples, we're talking about a huge geographical area, right? We expect that the vast majority of our samples to be negative, right? So to increase our throughput, we adopted a pooling strategy that we actually used during the pandemic. My lab developed a COVID, saliva-based COVID test using paired pooling, right? And basically what this does is we would arrange 96 samples on a 96 well plate. And then we would take a pipette tip and then we would create a pool, right, from this row by taking a bit of sample from each of these into this pool, right? That's one pool. If we use an 8-channel pipetter and we do that, now we can create 8 pools very quickly, right? That's one set of pools. Then we turn our pipette this way and we create a second set of pools by taking our pipette, taking up a sample and then turning it 90 degrees and depositing it in this column, such that we have 2 sets of pools where each individual sample is represented in one unique pool, right? So for example, if we had just one positive on this plate, it would be identified by by pool A being positive and pool 1 being positive. And it's the intersection of those 2 positive pools that identifies your single positive. In that case, technically, you do not have to run any additional samples from those pools. You've identified the one. Obviously, during COVID, we would confirm those. And if you have more than one positive sample on your plate, if you have 3, like shown here, you've got a lot of potential positives. So it's all these intersections that we have to deconvolute. right? So we adopted this for our RT-Quick assay and detecting prions because we expect very few positives, right? And that is shown here. So this is from Navajo Nation collection. We've got 64 samples. And to make sure that we could identify a positive in our pools, we spiked one of them, right? So we spiked this pool. And so we predicted that it would be the intersection of these pools that would identify the positive. So we predict that pools F and 3 would be positive, right? And that's exactly what we see here, right? So this is our graph showing you the amyloid formation rate that identifies positive samples. Here's our positive controls. These are a known CWD brain homogenate spiked into these samples. And you can see pool F and pool 3 are positive. That means that our assay is working, right? And so we did this for hundreds of samples in the Navajo Nation. Didn't see anything for years and years of sampling. And then we found one positive, right? And that positive was found in this area of the part of the Navajo Nation. And if I zoom in a little bit further, it was at the end of this canyon, Torrey Chinlin Canyon. And these are the actual fecal pellets that we found. One of the problems we have with these identifications is we think this is deer, but we can't rule out that it's sheep because there's a lot of ranchers on the reservation that move their animals through. So we are still trying to develop a way to determine what species these pellets are from, because that will determine whether or not it truly is a chronic wasting disease identification or a scrapie identification, right, from sheep. Okay, that is ongoing. Since that one discovery, we've found a few other positives, a total of 5, and those are shown here. The yellow targets and the red targets. The red targets means that every one of our replicates was positive, and the yellow targets means that a proportion of our replicates were positive. Okay, we have more confidence when we have 100% of our replicates positive. That's just our designation. Okay, so we do have some additional samples of interest that we are following up on and continuing to record our prevalence rates. Okay. So now on to the Rocky Mountain National Park. And again, we've been sampling here since 2017. And this is the overall area. Again, I just wanted to point out this Colorado River Trail collection is in an area where there's been positive animals found and no wildfire, right? And then, of course, we sample through this burn scar, and this is the blown up version of that, okay? And these are just some typical areas that we sample in and what the sample conditions look like. This is from Onahu Creek in 2020. This is part of that burn scar, okay? And that's before the East Troublesome Fire. And this is Onahu Creek area after. I think these are from 2021. These samples were from last season, right? And I put these here because you can see the charred soil. You can see the charred fecal pellets. I mean, they almost look like little charcoal briquettes, right? So we've sampled these and tested them for CWD using our RT-Quick and our paired pool strategy. And as you can see in 2020, before the East Troublesome Fire, we identified a few positives. We haven't identified a single positive in that area since, with one potential caveat I'll get to in just a second. This is that Colorado River Trail collection that I was showing you. And you can see here, we have a lot of positive pools, right? So this is where the pooling strategy breaks down. If you have a higher prevalence on the 64 well grid, higher than about 5 or 6%. That's about, 3 or 4 samples. Then a lot of your pools start to come up, and then the deconvolution, like, trying to test all of those pool intersections, you just have a uh, reduced efficiency, right? You end up having to retest a lot of individual samples. That's where we're at right now. But Colorado River Trail had a lot of positives. And recall, this area was never burned in any wildfire for decades at least. Okay, so here's the caveat. We were so excited, we've tested hundreds of samples, never found a single positive sample in a burn start anywhere. And last summer, I found these samples. They were, sitting on a bunch of charred wood and charred soil. I took them just like I did all of the others. They're very charcoal briquette-like. This was positive. This sample was positive. So we'll be confirming this. And what well, I'll get to this part in a second. We want to take that sample and burn it in our laboratory and see when we can sterilize it, right? And see what burn conditions are required to do that, okay? And that will complement what we're doing in this Arkansas study as well, okay? And I'll get that in a second. So again, this is the area that our collaborators are collecting in before the prescribed burn. It's in the Gene Rush Wild Management Area. Again, just south of the Buffalo River, just north, the Arkansas, the Boston Mountains are right there. And this is a blown out version, so you can see where it is in the state. It's in the northwest corner of the state. The Buffalo River is here. Oklahoma, Missouri are their neighbors to the west and the north. Also CWD endemic areas. And we were going to do pooled testing for these Arkansas samples, and at the last second, Annalise, my lab manager, decided not to, and thank God, because almost every one of these samples that they pulled out of that area have been positive. We have never seen anything like this in the Navajo Nation or the Rocky Mountain National Park, okay? We are trying to confirm this. A couple of our controls didn't work that great. So our NBH controls worked well. I mean, our NBH samples were clean. Our E2 samples, that's our positive control, didn't work on this run, right? So what we think is that these are true positives, but we can't rule out the ones that are negative as true negatives because our positive control didn't work. So we're trying to repeat this data and see if this rate of positivity is real. In the meantime, we are doing laboratory studies of burning and trying to mimic controlled burns and wildfires, okay? So in this experiment, we take a small 1-inch piece, it's about 10 milligrams of a stem from a plant. We dip that into a 10% infected brain homogenate with CWD. Right, we let that dry. We put those stems into these crucibles, and then we burn them at 650 degrees for 1 minute, 2 minutes, or 4 minutes. We take this, it's basically ash. It's not completely ashed yet, but it's very close. We resuspend these samples into 1 mil of extraction buffer. And then we compare that extracted sample to our control sample that was not burned, but was also extracted, okay? And this is what that 96 well plate looks like. We do all of these samples in octuplicate. And so the first 4 samples are a serial dilution of just the E2, the CWD brain homogenate that we use to contaminate our plant samples, not burned, but ran on the plate. And then we have our extracted samples, which we burned at those different, these samples are at 4 minutes at 650 degrees, and these are our control samples. this is really, really raw data, right? We just pulled this off yesterday. And so these are the traces you see. The RT quick reaction is sort of analogous to PCR. So if you've ever done real-time PCR, you see these types of curves where nothing happens. And then as you make your PCR product, it fluoresces. Same thing, but instead of making a PCR product, we're making an amyloid surrogate marker. Okay, it's not a real prion, it's not infectious, but it serves as a really good surrogate marker. So, as I hope you can appreciate, our dilutions for our dip that we dipped our samples in are still positive at 10 to the minus 6. When we burn the samples at those conditions, we lose our signal pretty much around 10 to the minus 4, right? Which tells us we've got about 2 log-fold reduction of prion titers after burning, okay? I have to tell you that we were kind of surprised that this wasn't completely eliminated, the prion signal, right? But there is one paper that did a similar experiment to this in 2004, right? But they used an even higher concentration of prions. They call it specified risk material, the brain and spinal cord from a hamster. And they burned their samples at 650 degrees for 15 minutes. And then they did a bioassay. So not an amplification assay like we're doing, but a bioassay. And 2 of 21 mice still came down with disease. So while they probably eliminated 8 logs of infectivity, there was still some infectivity remaining, right? So in light of this, they did burn a lot more material. They burned 1 gram of hamster brain. We were burning about 10 milligrams. We estimate contaminating those sticks. So we still were a little bit surprised that we were getting that robust of a prion signal after these conditions. So obviously, we're going back, we're going to burn for longer and see at what point we can eliminate this prion signal. Okay. We are actually trying to model this for not just controlled burns, but wildfires. Controlled burns burn at a much lower temperature, somewhere around 300 degrees centigrade. Wildfires burn 500 degrees centigrade and above, sometimes up to 2000 degrees centigrade. So we did do this experiment at 300 and 450, and we basically saw no elimination of the prion signal at all. Okay. So if you're not scared yet, even in a wildfire, now we've have evidence that prions could survive a wildfire. And we've even have one sample that we recovered from the burn scar that looks to be positive as well. So right now we're trying to repeat these results and then find conditions at which we do finally eliminate these signals. One other caveat I'd like to mention too, and that is the amount of prions that we're expecting to find on the landscape is going to be many, many orders of magnitude lower than this specified risk material that Carl Gajdzak used in his incineration experiments and in our experimentally controlled burns as well, right? So another part of this is to use a much lower dilution of infected brain homogenate to contaminate our samples with, to mimic more closely what we expect to find on the landscape. We tried to do that earlier, and it took us months to just optimize our extraction, right? So we do have the extraction really nicely worked out now, and so we're going to go back and investigate those lower titers and see what burn conditions might eliminate those lower titers as well. These experiments that I described today will help inform how we set those additional experiments up. Okay. So just to summarize, I just wanted to say, I didn't talk about this, but we did simplify a homogenization protocol for the feces that we were testing on the landscape too, and it really helped with our consistency and our throughput. The paired pooling really is a game changer. As long as we have a low prevalence estimates, we can run up to 320 samples per RT quick plate. The results from our Navajo Nation so far, we found 5 positive samples out of 378. Again, I must stress these are preliminary. This data has not been peer reviewed yet. And we still don't know if it's scrapie definitively or if it's CWD. Okay, so those aspects are ongoing. We also found positive samples in the Rocky Mountain National Park before a wildfire burned through those areas. So far, conservatively 3 of 72, although the Colorado River Trail, we undoubtedly have identified many more. And now we've got 1 positive sample found in the burn scar. So 1 out of about 300 samples that we've tested so far, okay? We also think that Arkansas has a lot of positive samples in that endemic area in Gene Rush. And that is prior to the controlled burn. So once we get with our collaborators and discuss the data, we're going to come up with parameters that they're going to try to use, and then we're going to resample and see if that rate of positive detection decreases. Fingers crossed. And then, yeah, so these controlled burn experiments that I was just describing today will help inform the actual on-site conditions that they're going to try to use on the landscape in Arkansas. So those are the 3 sort of vignettes I have about our work today. One thing that I didn't talk about, because I wanted to save some time for discussion, but I think it's relevant to the One Health concept, and that is I learned a lot of what I did not know about collaborating, especially with our partners at the Navajo Nation. And I have time. I want to share with you a little anecdote that is very embarrassing to me, but I think it's instructive for when you're working with communities, right? As a scientist and working with, Western knowledge versus, boots on the ground, traditional knowledge, right? So as I said, I tried to backpack through the Navajo Nation to collect samples one summer and almost killed myself because it was so hot and there were so few samples. And then my friend and colleague Gilbert John, who's a member of the Navajo Nation, came with me and tried to direct me to more samples, right? And so we got to a point where we couldn't drive anymore. So we got out and started walking, looking for samples. And I have my GPS out and I'm like looking at it like the Bible because, it's a vast area and it was extremely hot and extremely dry. And as we were trying to get back to the car, Gilbert was like, car's this way. And I'm like, I don't know, my GPS, it says it's this way, I'm talking to a person who grew up there, right? He was a boy. He spent his whole life there. It's like, Gilbert, I just don't know, It's like, no, I'm sure it's this way. And I'm sure you can guess he was right, right? And so it's a metaphor, right? I'm sitting there with my technology, right? And saying, no, my technology, my knowledge is telling me I'm right. And I was definitely not, right? That was a very humbling experience. It did not happen again. And Gilbert, not only working with Gilbert, but Gilbert working with ranchers and the elders in the Sweetwater chapter, of which he's a member, so gracious, and just so knowledgeable about the landscape. And we would not have been able to do this work without that help and support. So I just wanted to acknowledge that. Okay. And with that, I think we have plenty of time for some questions if you guys have them.
I have so many, but we can open up to everybody else first and maybe even hold down the slide so we can see if we get some online questions.
You mentioned taking different samples, but you never mentioned water. Is it prevalent in water?
Yeah, that's a great question. First, almost no water in the Navajo Nation to take, right? In the Rocky Mountain National Park, I mean, most of the sampling we do are in the riparian area, so lots of.
Your samples were along the water, a lot of the waterways.
Exactly, right. And so what we would do there is we would collect feces, we would collect plant samples that the feces were landing on. And then if there was any water within, 3 meters or so, we would grab a water sample. We would also grab soil samples, but this is interesting. For the first couple of years, we have to apply for permits, collection permits in the park. And for the first few years, they were fine with us collecting soil. And then all of a sudden they're like, oh no, you can't take any soil, you can't disturb it. And I mean, we're taking grams of soil. I can't imagine, what the problem is with doing that. So they wouldn't let us take soil anymore. However, when we pull up plants and sometimes the roots, whatever, and so it just wouldn't happen to find its way into our collection bag. But yeah, so we were opportunistic on the water. And we have not found a positive water sample yet. Having said that, published a paper 15 years ago looking at water from the Fort Collins water treatment facility before and after. And my graduate student found we can't definitively say it's prions because it did not cause disease in any of the animals. But by a similar assay, PMCA, we found PK-resistant water, PK-resistant prion protein in water before the water entered the water treatment facility. Didn't find it after. Like I said, we didn't call it prions because it didn't cause disease in an animal. So that's why. But water, yeah, is something that we're interested in.
Do you think a decrease in animal activity in the bird scar would affect the load you're seeing?
Great question. And we debate this all the time, right? Because on the one hand, if you're trying to use this for prion mitigation on the landscape, like a controlled burn, the question arises, obviously they're not going to frequent that area for a long time until the forage comes back. But another unintended consequence is, especially in wildfires, probably not so much in controlled burns, but in wildfires, those animals can be pushed in different directions. They can be pushed together. And if they're pushed together, now that increases direct transmission, which is, we know, more efficient than indirect transmission. So there's still a debate, whether or not, the wildfires are a good thing because they eliminate the contamination on the landscape, or does it facilitate additional interactions with animals after the fire?
Maybe follow up about the bird scar. Does it, like that one that was positive, was that sort of on the edge or right in the middle? And I'm wondering about like the extent of the heat and the burning that's occurring.
Exactly. So that sample was more on the edge. It wasn't in the center of the burn area where, it really looks like a big ashtray, right? And then you sift through it, and then all of a sudden you find a burned fecal pellet. This was it was closer to the edge. And that's a great point too, because those wildfires don't burn at a consistent temperature. So and for a consistent time. So yeah, one could imagine that on the edges, it maybe is more the intensity of a controlled burn of 300, 400 degrees Celsius. And it may not burn for as long as it does in the center, or completely as it does in the center.
And then maybe a follow-up question Beth and I talk about is, in wildfires, what role do you think they might play? So obviously potentially decreasing the load if it's burning hot. But what about aerosolizing and then transporting to other locations?
Yeah, no, that's a great point. That actually they tried to address in that Gash-Zizek paper, because they were trying to model how to eliminate specified risk material in a commercial industrial grade incinerator. So they were concerned about that same thing, if there was environmental contaminants that would escape the incinerator. So they set up a benchtop model of that and collected the air that was coming out of the incinerator and tested those, and those did not cause disease in any of the animals. However, again, going back to the variable temperatures, one could imagine aerosolizing or some ash or something that's not completely burned that could still have prion titers in it, and then it floats away into another area. I think that's certainly a possibility. I think I guess our hypothesis is that, again, the loads that you're going to see in the environment are low enough that lower fire temperatures might eliminate. If it's just 1 or 2 logs, that might be enough to eliminate it, right? Again, prion contamination is not going to be homogeneous either, right? If you've got a gut pile there, that could be a significant titer that, a less than complete burn might not be able to eliminate. So I would definitely not rule out that possibility of it floating away into an ash. Definitely. I don't think it would necessarily, I think it would still be attached to some bioorganic material. I don't think, I could be wrong, but I don't think we would have like truly aerosolized prions without it being attached to something else. Jim.
You probably said this and I didn't hear it, but what proportion of these studies were elk? Did you include deer like in Arkansas? And so do you get differences in response based on elk or deer? And I don't know much about that.
Yeah, no, great question. And I did not address that, but I can say that what we're seeing in the Navajo Nation are mainly deer. Could be sheep, right? That's our big debate. In the Rocky Mountain National Park, it's easy to tell the difference between elk, moose, and deer pellets, and we get all of them. And great question, Jim. I don't know that I know we have this data. I would say it's something like probably 70% elk, 20% deer, and 10% moose feces. But I can confirm that. But the vast majority of the samples we're collecting in the Rocky Mountain National Park seem to be from elk.
And other studies have shown what about the prevalence in those 3 species?
So typically it's higher in whitetail and mule deer than it is in elk. But once it's established in an elk population, it is hard to eliminate. But typically you do see higher prevalence estimates in the smaller cervids. And even lower prevalence in moose. They're more solitary animals. That's a good point too. It could be, behavior could dictate those prevalence estimates. Elk are the biggest herd animals, right? They tend to congregate in the biggest herds. Deer are more kind of family oriented, smaller, and moose are solitary. So that kind of tracks with their disease prevalence, right? And makes sense because direct contact is the most efficient way to transmit.
I had another question. Let's see. When did you collect the samples after the burn, roughly?
Yeah, so we collected they didn't let us in 2021 because of the instability, I think. So in 2021, we were collecting just in areas that were not burned. They allowed us to go back in 2022. So it was a good, year and a half, 2 years after the burn when we went in there. And I can tell you that there's very little cervid activity still in those areas, right? So you don't see a lot of fresh fecal pellets in there yet. So it's taking a while for the cervids to come back into those areas.
And then that one sample that you found was in ash. It looked like it had been burned.
It was, yes, absolutely. Yeah, not completely burned, but it was definitely in a pile of ash surrounded by some wood. And I don't know if you could appreciate, but there's also new plant growth coming out. We test that too. We were really interested to see if any of the prions have entered the soil. And then with new plant growth, are they taking that up and making it available to naive cervids that are foraging in the area? We haven't found any positives in that new growth.
Yeah, a couple more quick questions. One is regarding water and soil. So tremendous pollution factor, obviously. And but your assays are quite sensitive. They can detect down to how many logs dilution?
Yeah, so far about 8 logs.
8 logs. So that's and per gram of ice.
Yeah, it's a swimming pool. It's a
Is
lot.
it?
It's a lot, yeah.
Yeah, so if it's equally distributed. I have to test the whole swimming pool. Sure.
Yeah, I mean, that's the nature of it, right? Because it is an amyloid, right? It's an aggregate. So even if you drop it into the swimming pool, it's not going to disperse, completely through it. So it could be just at the bottom near the drain, and so, you could sample the whole swimming pool and never find it until you get down there, right? That is absolutely a caveat in what we're doing. That's exactly why we started doing the paired pool testing, because we expected very, very few positives. And that's what we saw in the Navajo Nation and essentially in the Rocky Mountain National Park. When we got those Arkansas samples, we have to reassess. I don't think we can do pool testing on those. There's just too many positives.
This question, I think I heard about that study that Paul Brown did with burning, but there must have been a lot of other studies. So what's the summary of those?
Yeah, that Paul Brown study is the one I showed you, right? It's
Yeah.
the only one published.
Really?
It's the only
Wow,
one.
that's amazing.
It isn't, yeah, yep. It's fertile, right? That's why we're in it. But yeah, it is literally the only one, and it's a different survey too, right? It's commercial, industrial incineration to get rid of the specific material that is in abattoirs and slaughterhouses and things. And we're looking at it for a different landscape, wildfires, and one of the few options you have to remediate huge geographical areas, right, is a controlled burn.
We try to concentrate water like we've done, when you're looking for Sudanicha, for example, getting a salt from the ocean. And if we concentrate it, we sometimes can't detect it.
Yeah, we have tried to concentrate some of our water samples, both with lyophilizing, but also this precipitation assay or technique that preferentially pulls down prions using phosphotungstic acid. So we've concentrated those and still didn't have any positive samples from that. Another thing that we're working on now is working with Candy Mathiasen and Nate Bankers. He has this really cool iron oxide bead extraction that helps concentrate and really eliminate inhibitors. And so we're right now trying to optimize that and play around with that a little bit just to see if we can concentrate any signal from our other water samples.
Did you find any first droppings after the burn scar, after the fire in burn scar?
We have found a couple, but like I said, very few compared to, the rest of the park. I don't know if you guys have probably hiked through there, but if you ever, next time you're in the park, just look down, right? Every 2 feet, you're gonna step on a pellet, right? Nothing like that after the burn, right? I think we've maybe collected less than 10 fresh fecal samples from those burn scars.
To your sheath question, have you ever tried doing like cytochrome B on those pellets?
No.
So that's what we do with bat guano in the field when we're trying to figure out what bats they come from. And works.
So you can identify cytochrome C is different in different species.
We have to, exactly, as long as the sequences exist in GenBank, which I assume they do.
Yeah.
So that's what we would do. We would do either cytochrome B or COI.
Okay.
So there's two different assays depending on what sequences are available. And then you can identify the species that the feces came from.
I'll look into that. The one thing that we did find a couple of papers that have a protocol for PCR of feces. And we haven't tried it. Our hesitation there is the samples that we're pulling out of the Navajo Nation are so old
Yeah.
and dry
Yeah.
that we're not very hopeful. The other thing we're considering though is working with Jessica Metcalf to speciate the feces based on the microbiome.
Yeah.
That seems like a lot of work, but obviously this is an important question to answer, right?
I'm happy to share the protocol for you. You're welcome to this stuff.
That would be great. Yeah, we'll try anything.
Yep.
Yeah. I can say one thing that we did try was to use our amplification assay with a different substrate that's supposed to preferentially only amplify either deer, CWD prions or sheep stray prions. And it did differentiate. So our controls were, a CWD prion in this one substrate that's supposed to be exclusive for deer amplification, and they amplified just fine in both substrates. So we were hoping that would be the magic bullet, and it wasn't. So we need something different. So yeah, I will follow up with you about those protocols. Thank you.
Yeah.
Be great.
Any questions online?
I didn't see.
No.
Weird question. I guess it might be useful since some of these burn scars are really huge, like Cameron Peak, 200,000 acres.
Yeah.
So a person can go in and look at prevalence a few years later and just see if there's an effect that way.
Yeah, great point. And I think that's what we're trying to do in the Rocky Mountain National Park too. As you can see, all the collections we do are environmental samples, right? I'm not a wildlife biologist. I don't shoot a dart gun. I don't shoot any kind of gun. So I need help, right? So I would need, the NPS, I would need the Rocky Mountain National Park and the wildlife biologist there. But I might be able to convince them if I show them some data, right? To say, it might be worth to look at prevalence estimates now that we're, 5 years or so after the East Troublesome fire. I think that's a really interesting question. Anything online? Okay, thanks for joining us today.
I was gonna say, thank you so much, everybody online, and in person, and it's so great, and again, thank you for the importance.
My pleasure. Thank you for coming.
Oh, Cheryl,
Oh, Cheryl.
did you want to come off mute? Go ahead, Cheryl, we see your hand.
But you are muted.
Or if you want to put it in the chat.
Oh,
You have to,
Unmute me.
okay. Thanks, Cheryl.
Here, we'll have to check that setting next time.
You can't.
It's not letting us, Cheryl. Do you want to put it in the chat? That's so weird. We'll have
That
to
is
look at
weird.
that for next time.
Because they're external. So maybe it has to do with that.
Oh, we're gonna ask about deer carcasses. I think she put it in the chat. About deer carcass sampling. Yeah, great question, Cheryl. I was kind of just explaining that to Jim, at least for the Rocky Mountain National Park, where we would love to do that, and hopefully we can do some actual testing of the animals themselves. For the Navajo Nation, we have participated with the Navajo Nation Division of Wildlife to do some live animal testing. I think we've gotten some carcass testing from some hunters as well. That was a few years ago. And we haven't done that on a consistent basis. But the animals that we have tested, both live and the carcasses, were negative so far.
Thank you for pointing out our issues. We'll see if we can solve that for next time.
Yeah.
More
When-
about that, Cheryl.
Understand there are testing stations in some states. Yeah, great question. And the hunting station, the testing stations you're referring to, that's exactly how we got those samples, right? So we partnered with the Navajo Nation Department of Wildlife, Division of Wildlife, for getting samples to test from those testing stations. So that's exactly how we were able to get some of those samples. Thanks for your question. Okay.
All right, well, thank you again.
All right. Thanks, you guys.
Yeah. This is very good.